So hello everyone, thanks for coming to the seminary. So we will start now and go from here.
Okay, so bonjour, thank you all for coming. So today we have Francesc Piferrer from the Institute of Marine Sciences in the CSIC in Barcelona, in Spain, and he's the head of the group of biology of reproduction, and he has been working on development of sex and epigenetics for many years, and he's an expert on this field, and he will be doing a talk on epigenetics as a driver of adaptive evolution.
Bonjour, merci beaucoup, Nouria. Un plaisir pour moi d'être ici. On va parler en anglais parce que je suis habitué à parler anglais, not en français, en mon conférence. Merci beaucoup. So I'm going to talk to people who work in the Institute of Evolutionary Biology or Evolution about epigenetics as a type of adaptive evolution, although this is not my major field. No? We work on other aspects. In our group, what we do is to work on, I know that it's not correct maybe to say the difference between basic and applied research, but if we can put it this way, we work on basic research, especially on sex determination, things related to epigenetics, sex determination, transgenerational effects, methylation reprogramming, ploidy level compensation. We just published a paper came just yesterday, gene expression regulation, and then on a more applied aspects to especially to aquaculture, but also to fisheries and to monitoring the effects of climate change. We work on sex control markers and prediction, growth performance, disease resistance, all these things that are important for aquaculture. And then for fisheries, a very important aspect is age determination by the development of epigenetic clocks to measure or estimate the age of the fish. And then finally, in application to monitoring the effects of climate change, using a certain type of epialleles called metastable epialleles. And what we do is to work with different types of models, mostly the European sea bass, but also other fishes. And when it's necessary, we go back to published data, for example, on fugu, the model, or frogs, or even humans. So if we take a look at the concept of epigenetics as a driver of adaptive evolution, and this is outline that was put by the late Gunter Vogt in this book that of last year, Epigenetics in Fish Evolution, which is a very nice book. These are aspects that are important to understand epigenetics as a driver of adaptive evolution. So here we have ability of epigenetic mechanisms to produce phenotypic variability, and how we do that. Then this is due to the concourse of epigenetic mechanisms, and then also understanding what are the sources of epigenetic variation. Then we have epigenetic variation and associated phenotypic variation in natural populations, something that is very important, but we don't have, we are not touching that. Maybe some of you are doing, or Nuri, I think that is doing that. And then the autonomy, another important aspect, the autonomy of epigenetic variation from genetic variation. This is a key and also key concept. And then we have the stability, selectivity, and fitness effects of what these mutations do, and what are the corresponding phenotypes, and important fitness changes in epigenetically determined phenotypes. And then another very hot topic is the transgenerational inheritance and genetic integration of mutations. So, and then finally, different ways of how epigenetics can contribute to adaptive evolution. But of all these things here, we are going to talk about the ones that are put in the book. So the talk today is going to be centered in the aspects that in one way or another, we have touched a little bit, which is ability of epigenetic mechanisms to produce phenotypic variability, diversity, stability and selectivity and fitness effects, and then the transgenerational inheritance. So if we go to the first one, let's first, I'm sure that you all know very well this, but in case that there is somebody that is not familiar with this, let me explain briefly what we understand for epigenetics and the epigenetic mechanisms. So the concept here is that the epigenetics is the study of phenomena and mechanisms of both things that cause chromosome bound. And this is very important to have into account, because from the epigenetics point of view, the external environment is everything that is just above the chromatin and just above the chromosome. So it doesn't matter that whether you're talking about the external environment to the organism, to the internal environment of the organism, or even the internal environment of the cell, or even of the nucleus. Everything from the epigenetics point of view, environment is outside the chromatin. Okay, and the thing is that these changes can be mitotically and meiotically inherited. So not only during cell division, but also between generations. And since genetics deals with mutations, permanent mutations in the DNA sequence, epigenetics deals with alterations. And here, this makes possible the fact that each person or each individual, so we have, each one of us has a particular genome, and then that this genome, through these alterations and modifications, is able to give rise to the more than 200 cell types that we have in our body. So we have all the same genome in our cells, but different epigenomes depending on the cell type. Okay, so then the three major epigenetic mechanisms are DNA methylation, which is the addition of a methyl group, CH3, in a mostly in vertebrates. I'm talking vertebrates and invertebrates, but there are other types of methylation, but the most important one is methylation of a cytosine that is before a guanidine, so it's called a CpG, okay? And this creates this modification from being non-methylated with the CH3 group to be methylated. It's what we call epialleles. So they are genetically identical alleles, like in genetic alleles, but in this case with distinct epigenetic modifications. And this thing occurs directly into the DNA methylation in the DNA molecule. But then we have histone modifications. What are modifications in the histone tails? Many different types of modifications, acetylation, phosphorylation, methylation, and so forth. And then the concours of different types of non-coding RNA. And one of them, very important, is long non-coding RNAs, or for example, microRNAs. But my talk today is going to be centered mostly or exclusively on DNA methylation, which is the most genetic of the epigenetic mechanism because of its intimate contact with the DNA molecule. So, and in the same way that you can write a text and copy a text and erase a text with a pencil, so exactly the same thing, it's how it works, the epigenetic modification. So here we have, so this line here, simply, this line represents any DNA sequence, and then we have here sites that correspond to CpG sites that are not methylated with the concours of the DNA methyltransferase 3. Then we have this DNA sequence that becomes methylated, and this is called the de novo DNA methyltransferase, the number 3. But then once this is methylated, during mitosis, during cell division, then how is this copied? So this is copied by another enzyme called DNA methyltransferase 1. And then, so that's the way of how these methyl marks are written and are copied in the genome. And then we have two ways of losing these marks. One of them is active methylation, so like the erasing, like an eraser, and this is thanks to the enzyme called 1011 translocase. And then we have passive methylation, which is when during cell division there is inhibition of the maintenance methylation enzyme, the DNA methyltransferase 1. If this enzyme does not work, then simply by dilution, the mark is not longer present anymore. So this is the way how epigenetic marks are written, copied, and erased, okay? And this can be induced, these changes can be induced by many, many different factors. We want to see them. But just before I go to that, again, this is how it's represented, the sequence, just the simple line. The dots are places where the CpGs are methylated and the open dots are not methylated. And typically, and the dogma is that during gene expression regulation, when you have a promoter that is methylated, so it's a premethylation of the CG sites in the promoter, then the gene is off. When this doesn't happen, then the gene is on. This is the dogma, but this doesn't happen in many, even in other places like invertebrates or even within vertebrates. So This particularly takes importance in what is called the CPG islands. CPG islands are places where there is a high concentration, more than expected, of C-sheet nucleotides that can become non-methylated to methylated in the promoter region usually. And when there is no methylation, the gene is expressed. When there is methylation, the gene is not expressed. But what I was saying just a moment ago is that the dogma says that usually there is an inverse relationship between the level of DNA methylation and gene expression. So the more DNA methylation that you have, then the least gene expression. But in this study, we have shown that there is both negative, as you would expect according to this dogma, but also positive regulation. So it is not straightforward always how it is the relation between the two aspects, not DNA methylation and gene expression. And this is something that needs to be taken into account to interpret the results of your experiments. So, and to finalize this introduction, simply say that when you have two groups, for example, like here, like fish exposed to low temperature or fish exposed to high temperature, then we have different CPG sites. And depending on the level of methylation in one of them, for example, this is 3 out of 10, so 30%, same thing here. This is 70 or 60%. So then when there is a statistically significant difference or a difference that you establish in a threshold that you arbitrarily set, we usually use 15% differences in order to call something that is differentially methylated. Some people use less, some people use more, but in our hands, 15% is good, but then at the statistical value of P001. Okay, so then when this happens, then we can call differentially methylated cytosines, because this cytosine is differentially methylated between the two treatments. But as we say, one flower doesn't make a spring, so you need many flowers to have a spring. And it is more important to have, or it's more physiologically relevant to have what it's called differentially methylated regions, which is simply a collection of DMCs together in a given region. But I should say that there are cases that, yes, that one flower makes a spring because, for example, in one fish, the olive flounder, there is one cytosine, one CPG, the methylation of which implies whether this animal is able to sexually mature or not sexually mature. So sometimes one single CPG makes the difference between one phenotype and another phenotype, one single CPG. But if you can work with DMRs, it is better, I think, than to work with a single DMCs. No, but sometimes the DMCs are very useful too. And the way that then this is represented, you have different treatments here, and then you have different groups, and of course the level of methylation can be expressed in a percent, so going from 0 to 1. This is a typical value around 70, 80% or something like that in most vertebrates, and then that's how you express the data. Okay, so let's look at the source of epigenetic variation. So as I said before, we have different factors that are internal. The genetics has an influence on the epigenetic landscape. The tissue, very important. Tissues, perhaps one of the most important. Then physiological conditions, sex, differences between males and females, and then the age, which is the basis of the epigenetic blocks. But then we have also external factors like the diet, abiotic factors, exercise, pollutants, biotic factors, and so forth. And the thing important to have into mind is that epigenetic variation here depends on both. Depends on the genetic variation that conditions this epigenetic variation. So here is the influence of the genetics, and also of the environmental variation here, anything that is in this site. In this site, or even in this site, because this, as I said before, the physiological condition is something internal of the individual, but from the point of view of the epigenome is external. So these environmental influences through epimutations, so changes in the pattern of DNA methylation, for example, or histone modifications, then the two together give rise to epigenetic variation. But then there is a third source, which is a stochastic variation. And this is something that sometimes is not enough well taken into account, but is important to realize. And then both genetic variation here and epigenetic variation, they condition both gene expression and therefore the phenotypic variation. But as you can see here, this arrow has two ways, no? It's a two-way arrow. And this means that also gene expression also is able to condition epigenetic variations. That introduces one aspect very important, which is an epigenetic variation is the cause or is the consequences. Yes, the epigenetic variation is the cause or the consequences of a certain pattern of gene expression. So sometimes when genes are no longer needed to be expressed and they are shut down because they are not needed for that tissue, then methylation changes is a way to stabilize the no expression of that gene. Okay, so it can work in both ways. All right. So if we move to a little bit to talk about most of the talk is going to be at the end here, but if we look at the fitness changes in epigenetically determined phenotypes, here I would like to say that a concept that is emerging is the concept of epigenetic traps. And here we have two different types of environment during early development and during adult. And here we have during early development, for example, a cold temperature or a high temperature. And then there is the temperature that the individual finds when this individual is adult. And if is exposed to, let's say, this certain condition and then finds the same condition later, then there is no problem, it's a match. Or if it's found in high temperature and then finds high temperature, it's also a match. The problem comes when you have like a certain temperature during early development, which is able to program the epigenome in a certain way, and then you have a different temperature. For example, you go from cold to hot or from hot to cold. Then there is a mismatch, and this sometimes can produce an epigenetic trap. And these epigenetic traps are changes that arise in response to a novel environmental cue that produce maladaptive phenotypes if that phenotype that produces doesn't confer any advantage to the organism that suffers this. This is something that happens to nature, and in like mutations, most of the mutations induced by the environment are neutral or maladaptive. There is no positive. There can be, of course, no, like normal mutations, but most of the mutations are neutral or maladaptive, not positive. Okay, but the same thing that is bad in nature can be good in culture. So in culture, this, of course, there is a lot of debate on how the sex ratios can induce changes in sex ratios induced by temperature, in which we know that the epigenetic mechanisms are very important, whether this is maladaptive or not, and so on. No? So the epigenetic traps in relation with sex ratios is something that is being very debated and a hot topic nowadays. But what I was saying is that in culture, this can be exploited to advantage. For example, in agriculture of the Chinese tongue sole. This is a species with a high sexual size dimorphism. The females are much bigger than the males. The females are ZW, the sex determining system. The males are ZZ. At normal temperature, 22 degrees, a male is a male and a female is a female. But then if you put a female at high temperature, and then this animal, this ZW, still is a ZW, the chromosome does not change, but it becomes, because of the effect of temperature, a male. And this is called a neo-male or pseudo-male. Okay? The thing is that when you have this pseudo-male and you use this pseudo-male to fertilize normal females, then this animal produces offspring that also has a high rate of sex reversal, even when you produce them at low temperature. So the temperature-induced sex reversal in this guy has been transmitted to the next generation and expresses this sex reversal phenotype even when you culture them at normal temperature. And this is something that this epigenetic trap is something that can be bad in certain circumstances or mostly in nature, but can be exploited in aquaculture. And one of the ways to exploit this, for example, and what we're going to see now briefly example in nutritional programming in aquaculture. So in a recent paper published by Nuria, then here what we did is to expose the different groups of fish at in this case at the F0 here to either control in white or in black high temperature. And then we did the same thing at the F1. So we created first 2 groups and then 4 groups. And then we named these groups control, and then control treated control, and treated temperature. Okay, so these are the 4 combinations. And when you look at the sex ratio of the F1, then you see an increase in the number of males. This is what you would expect. But in terms of epigenetics, the nice thing about this paper is that for one of the very first times, tries to classify the epigenetic changes in different categories. And here we have different possibilities. No, we're going to start from the top or from the bottom. But for example, if we start from the bottom here, if there is no effects at the F0 in terms of epigenetic changes or at the F1, and there is no interaction between the 2 temperatures, then we say no effects whatsoever. But then we can have, for example, no interaction and significant effects at the F0, but not at the F1. So we have F0 effects that can be in one way positive or negative. The same thing for the F1. And then if we have 2 effects but no interaction, then we can have what we call additive F0 plus F1, same direction. But these 2 effects can be in opposite directions. So then the F0 is different from the F1. So this is opposite direction. And then we may have not significant or significant effects in the F0 and the F1, and also interaction. And then it's called quantitative interaction, same direction. Or we have also interaction, effects in both places, and a qualitative interaction opposite directions. And here, when we look, for example, at certain parts of the genome, either in females in this side or in males in that side, we see different types. These are the 4 groups, the 4 treatment groups here. And this is the amount of percent methylation of each one of these loci, of these places in the genome of these groups. As you can see here, there is an additive effect. So the higher the amount of temperature in different generations, there is an increase or a decrease. The same thing for females and males. No? Here there is a quantitative effect, no, in this way or in that way. So like a U in this way or that way, and then a qualitative interaction in this pattern. No? And this now we see that this matches in a paper that still is not published, but now we see that some of these epigenetic changes matches the sex ratios. So we see now, we have found now that contrast to what you would expect, that when you deviate too much sex ratios in one given temperature and you keep giving this temperature in different generations, then there is like a bounce back effect, no? Something that somehow matches what we found here in this effect. And this, in addition, depends on the genetic background. So to put it simple and to make it, I'm sorry, very understandable, it's like these systems that are very boring, very annoying for me in the car, when the car like stays on the lane, no? This automatic control system, no? The more that you try to go out, the more that the car corrects back in, no? So we found exactly this. The more that you try to deviate from the Fischerian sex ratio 1:1, the more the system seems to bounce back, no? It would be nice to see, but this is difficult, to see what would happen in the F3, but unfortunately, we don't have any more the fish. No? This experiment that I'm talking now about, it lasted more than 10 years to do. So because for the sea bass, it takes different generations. So okay, so let's look now at transgenerational inheritance and integration of epi mutations into the phenotype. So when we talk about epigenetic inheritance, here we have the life cycle of a typical individual, a fish, a crustacean, an oyster, whatever. And here we have three shaded areas. These three shaded areas means the windows of sensitivity in which the animals are more sensitive in terms of epigenomic reprogramming to the effects of the environment. So the most sensitive part is during early development, during the formation of the zygote, embryogenesis, until hatching. The second most sensitive part is during gametogenesis, the formation of the gametes, sperm or eggs. And the third, if present, because not everybody has metamorphosis, but the third part is during metamorphosis, okay? So if we look at this part here between when the animal is about to be born, fish, egg, for example, embryo, late embryo, until it becomes an adult, any change in the environment that is able to affect the epigenome is what we call context-dependent epigenetic programming. And this in aquaculture or animal production in general, it has practical interest. And this is based on the inheritance of epigenetic marks during mitosis, during cell division. So here we have typical like DNA with the histone modifications. And for example, here we have one mark, histone mark or DNA methylation or whatever epigenetic modifications you want. And then during this differentiated cell, and then in germ cell, and then this or somatic cell, and then this during mitosis division, then gets replicated into the daughter cells. So anything that happens here at the very beginning then is carried around in the daughters of these first cells that were affected by the early environment. That's why it's important, this early environment. Of course, here, the effects here, in this part, the effects are higher here and then progressively washed out here. And this has two types of different consequences. If when this, whatever environmental effect affects in whatever manner the epigenome, it's very different whether the tissue and the organs that are targeted or that are affected are undifferentiated or differentiated or already finally differentiated. So if the tissues and organs are undifferentiated, then what these epigenetic changes can do is to control or to determine the cell fate. So for example, if this happens in an undifferentiated gonad of a fish, is able to change this gonad either from going to a female that was supposed to be according to the genetic background to a male. No? That's the origin of a new male. And so in this case, we're talking about cell fate commitment. But if these epigenetic changes catches a cell or tissue and organ that is already differentiating or completely differentiated, then it's a more fine-tuning of the transcriptomic and the genome of this tissue. And then we talk about nutritional programming or the use of prognostic biomarkers. Why? Nutritional programming is based on the fact that, as you may know, in aquaculture nowadays, many carnivorous fish, like the sea bass or the shrimp, for example, are given diets that are vegetable diets. And this is not good because the animals, the fish, are very poor in carbohydrate digestion. Okay, so they don't like to very much carbohydrates because in the natural diet in the wild we don't have basically carbohydrates. And then if you feed them with a plant-based diet, which now is the tendency in aquaculture to avoid the use of fish oil and fish meal, then they become inflamed. How to solve this? You program the fish in order to make them resistant to the effects of to tolerate the plant-based diets. And how do you do that? You feed them with a plant-based diet at the very beginning. You lie to them. You tease them. You make to think that that's what they're going to find. You program them, but without allowing them to reach inflammation or anything like that, then you put them in normal diet and then let them grow the digestive system and everything. And when they are juvenile adults, then you feed them with a substitution diet. And this works much better if you did not program them at the beginning. And this is based precisely on the effects at these early stages. Okay? So if we look now at the most important, perhaps interesting thing is when you deal with the other side of the cycle, during gametogenesis and fertilization. And this is what we call germline-dependent epigenetic programming. And here we have the germ cells, the gametes, but again, something that sometimes is not well distinguished. Here we have to distinguish whether the effect is in the cytoplasm or in the nucleus. If it's in the cytoplasm, for example, we talk about nutritional programming in the way that we program the fathers to make better offspring. And how we do that? Through the cytoplasm, by looking at the cargo of proteins and non-coding RNAs and microRNAs and stuff like that, that these eggs will carry and to make the next generation, the zygote and the next generation. But if the changes, epigenetic changes, are in the nucleus, then we talk about intergenerational inheritance, truly. And this can happen across 2 or more generations. So in this case, we go from the F0 to the F1. So if we have an external factor that will affect the epigenome of the individual and the germ cells, then the F1, of course, will be affected. This guy derives from the germ cells here. But then if we see the effects in the F2, which have not been exposed to the initial, then we can talk about truly transgenerational epigenetic effects. Here can be epigenetic or can be confused with a maternal effect. So what are the advantages of aquatic animals to study epigenetics and to apply epigenetics? Well, the thing is that here, the epigenetic marks, when you produce the sperm and the gametes, there is one process called reprogramming, in which is like resetting a computer. It's like formatting a computer. You erase everything and you allow everything that happened to your life, all the epigenetic modifications and blah, blah, blah. Then it's sort of like putting a blank sheet to start a new individual again. And this in mammals is very clear, because in mammals, which are the 2 red lines, the mouse and the human, there is a lot of reprogramming. In fish, at the beginning, it was thought that this was the same. No? These are in blue. We have data in fish showing that there is a lot of also like in reprogramming in mammals. But now we know that this data is not reliable. And this is a technical issue with these experiments. These have been published, but this is not reliable. Why? Because they use a plate-based system, which is not single nucleotide resolution, and you cannot determine very well the results. And when in the same species, you use a single nucleotide resolution technique, more fine, we see that in fish, there is reprogramming, but very little. So you can see from 85 to 78 or something like that. So it's very little reprogramming. What this means that in principle, in theory, it means that any change that happens to the fathers has, in theory, more chances to be passed to the next generation in aquatic animals. And on top, here, if we compare the fertilization development in mammals, everything is internal. In birds, fertilization still is internal, but development is already external. And in aquatic animals, well, no matter whether it's mollusks or crustaceans or fish, everything is external. So therefore, in aquatic animals, it's very easy and very nice to a very good organism, not only to study epigenetics, but also to try to apply this knowledge. No? And this was the product of this book, in which we talk about many authors about the mechanisms, the applications, and biomarker discovery and stuff. Okay, so what amount, you can ask yourself, what amount of these environmentally induced epigenetic marks are transmitted to the next generation? And this is exactly what we try to answer with this experiment also by Nuria, and the colleagues at Piferrer in Palavas. So here, the same thing. So here we look at the two different temperatures here, different sites, different site 1, 2, 3, 4. And then we created these 4 groups in duplicate, and then we created still 8 groups and so on. No? So each group has a 3-letter code, meaning control, 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 and so forth. No? Control, control, treated, so forth. No? So we have these 8 groups here. And to make this story short, long story short, here what we try to do is to see, okay, if we have induced an epigenetic change here, then this data is what shows is that what happens in here. No? So the individuals that have been exposed to temperature in the parents, but not the offspring. No? How many marks? And then there is different control groups, the generation of 0, unexposed fathers. The exposed fathers, you see changes in methylation in places where they don't have it in the unexposed fathers, and then this is carried down in the daughters. This specific example is transmission from fathers to daughters. Okay, and how much of this environmentally induced epigenetic changes are transmitted? Well, it's about 5%, 4% in one sex, 6% in another, average about 5%. And this is a lot or nothing. I think that 5% for this type of thing is something, I don't want to say that is a lot because 5% doesn't sound a lot, but I think that is remarkable that 5% of the environmentally induced changes can be transmitted in a generation. Of course, now we are going to look at the F2 and see what happens. No, it's a big mystery what's going to happen in the F2, especially because between the F1 and the F2, and this is something that is not taken very often into account, is that it's not the same to create the F2 after 1 round of spermatogenesis or gametogenesis that after 2 rounds, 3 rounds, 4 rounds, because there may be what is called epigenetic washout. So the marks can be washed out at any round of spermatogenesis or gametogenesis. So this is another variable that is important when you work with oysters, with mussels, with whatever you want to work. Is something important to take into account. But one thing that I can tell you is that are these marks, no matter what, important for natural populations? And maybe here for the institute that you are working, you put your ears like that, like what's going to say now. No? Well, what we found, and this is something that we could found in cold-blooded vertebrates, not in hot-blooded, because in hot-blooded vertebrates, sex reversal only has a genetic basis, but in cold-blooded vertebrates has both genetic and environmental basis. What we found, this is a literature review, but what we found is that in the places where it has been studied, no matter whether you talk about fish, about some lizards or amphibian, we found that there is a difference in the susceptibility to sex reversal. Like the same thing with what we saw before with that fish, because the same thing happens, but not under agricultural conditions, but in the wild. These are experiments, or these are experiments. These are data from wild experiments, and from wild captures and from looking at the wild populations, not under different conditions. And here is the strength of the environmental stressor, for example, temperature. So this is a temperature 2 higher than temperature 1. And what this graphic shows is that the strength to achieve, for example, 50% sex reversal, you need a higher temperature in non-sex reversal, in animals that are the sons and daughters of non-sex reverse parents. But if you are the son or the daughter of a sex reverse parent, then the chances of you becoming sex reversal spontaneously without any effect is higher. And this is something that is quite relevant in the context of global change and so forth. And this is exactly what I told you before in the experiment that I mentioned before in the experiment of the tongue sole, no? That this can be exploited now in aquaculture. Okay, so there is a tendency also in natural populations, not in the one that from the experiment I just showed you, to find this. And this brings me to this concept that I was mentioning before, that is something that you have to take into account. No? the transgenerational washout of the epigenetic effect. It's not the same thing that you induce the epigenetic effect here and the F0, always not by nomenclature. And then this can be stronger, but can be progressively diluted in different generations. This is something that, that's why I was saying that for us, the F2 generation, that we had different rounds of spermatogenesis was complicated. And on top, not only the generation, but the number of rounds of spermatogenesis that you use to create one generation, or you wait to create one generation again. But then there is more. Then there is the threshold that you use to study how much you have an effect. No? So here, imagine that you have the magnitude of the phenotypic trait here from nothing to a lot, and then this is at different generations. If you have a detection line here, you will say that, okay, you see you have the effect here in the F1, you have the effect 2 in the F2, and then you will say, using this threshold, that you don't see the effect in the F3. But if you lower your threshold, then you will see the effect in the F3 and even in the F4. So bottom line, the thresholds that you use to determine the effects of the changes are very important. But not only that, this is the average of a population, but the population is made of individuals, and individuals may respond in different manners. So here you have 4 individuals in the population, or 5, and then these individuals are exposed to the F0 to a temperature or whatever that changes the genome, and you create a magnitude of modified epigenetic characteristic or the trait. No? But there are individuals that are very resistant and they keep going for a long time, but individuals that, of course, are very labile and they lose the effect immediately. So depending on how you look at that, you may conclude that these individuals, that if you look at only these individuals, you will conclude that the effect is very short, but if you look at these individuals, you will conclude the effect is very large. So here the message is that it's very important to take into account differences between individuals of the same population. And this is in this book that was published last year too, I mentioned before, the Epigenetics in Fish Evolution. This is the typical setup for a study environmentally induced epigenetic changes across generations. So you have a genome that doesn't change, with a certain epigenetic modifications in the natural environment. You put these animals, they have F0, in the altered or controlled environment. You expose them during early development. You may induce a change the epigenetic profile, and then this is induced during early development and is maintained during adulthood. And then during different generations, some of them are maintained and some of them are lost. But the important point is that some of them can become assimilated eventually as genetic changes. And that's what I'm trying to talk now in the next slides. The next slides, I'm going to be finishing with this basically. In the next slides, I'm going to talk about domestication and how domestication is a very fertile ground to study epigenetics. Because domestication means basically to take an individual and a species, put it in a completely different environment. So Charles Darwin in 1868, when he wrote the book on the variation of animals and plants under domestication, he mentions in his book that there are a certain amount of changes in domesticated animals that are losses of pigmentation here, floppy ears, not the cats, because the cats that you have at home are basically a wild animal, but the dogs are much more domesticated than the cats. And that's why many dogs have the floppy ears, or the horses, or the pigs, or whatever, compared to wild boars, no? The floppy ears, they fall down. And then you have an altered snout, which means that shorter snout, curly tails, smaller brains, and especially very tame. That's what you select for. And this is called, nowadays, he did not put this name, Darwin himself, but nowadays this is called the domestication syndrome, this set of characteristic things. But then in the 19th, and of course, when nowadays you try to compare a wild animal with a domesticated, what you are doing, in fact, is comparing two different species because they have been exposed to selection for thousands and thousands of years. So when you compare a wild boar with a domestic pig or a gray wolf with a domestic dog, you actually are comparing the result of this domestication process. But interestingly, in the 1950s, this guy, Belayev, in Siberia, he was sent there because he was Spanish and sent to Siberia. So he started this experiment on the domestication of fox. And what he did is to select foxes, white foxes, only exclusively for tameness. Okay, so to be Berosal didn't care about the color of the hair, about the growth, about anything else, but just tameness. And what he found is that in selecting for tameness, he was actually replicating all the aspects of the domestication syndrome, becoming even more wider, floppy ears, and blah blah, these kind of things. And then Wilkins, the editor of genetics, in 2014, they proposed that these effects that happened during early domestication are due to that the new environment, what is actually doing is affecting the cells of the neural crest. And if you remember, when you were studying biology, you were told that animals have three embryonic layers: ectoderm, mesoderm, and endoderm. This is true for all the animals except for the vertebrates, including us. We have what is called the fourth embryonic layer. And the fourth embryonic layer is not a layer itself, but it's called the fourth embryonic layer, are the set of the neural crest cells. These neural crest cells are the green ones, and this is the future ectoderm, the epidermis. This is the neural plate border when it falls and then forms the neural tube. And then these cells here in the place where it falls, they become the neural crest cells. And neural crest cells are very interesting because they are the cells that they give rise to certain cell types in the body. So, for example, all our melanocytes make us be more brown or more tan, all these cells of the body derive from the neural crest cells. And then there is another thing: our bones are derived from the mesoderm. All the bones except the bones of the face, of the maxilla and the mandibula. These bones are derived from the neural crest, all of them. Okay, this part of the face. Listen, here we're talking about pigmentation, right? So we're talking about melanocytes. Here we're talking about altered snout. No? So we're talking about all this part of the face. So, and if you look, in fact, in many farm animals and farm fish, it's very common to have this phenotype. So you have problems, this is problems with prognatism in the alterations in the lower jaw. So what we did is the hypothesis is to ask whether the first stages of domestication and adaptation to a new environment likely involve epigenetic changes. So if the epigenetic changes are able to capture and to transfer environmental information to the genotype and then to integrate this into the phenotype, is this possible or not? And what we did in this experiment is to compare wild fish with early domesticated, so only 1 or 2 generations of domestication. And then we compare the genetic differences, gene methylation, and gene expression. And what we found is at the 4 embryonic tissues different from the 4 embryonic layers, we found, if we zoom in any part of the genome, we found changes all over the place. And then we found for each tissue changes in gene expression. And then when we look at the GO terms, we look at the developmental process and so forth, and neural crest. Okay, so here I start appearing the neural crest. And then when we look at not at the problem, but at the very early development at the X, we saw that in average is different from tissues, no? This is the absolute value in green, and in red is the relative number in proportion. So the average of all the 4 red columns is this dotted line, just about 20%. So that means that 20% of the genetic changes that we see in yellow are already present at the time when gastrulation starts, when all the different organs start to be differentiated. And when we look at the GO terms associated, what we found very interestingly is that everything is like mesoderm, neural crest, ectoderm, neural tube, notochord, everything a lot related to the neural crest. And then here we have some examples. Here it is the Adams T9, and here we have the levels of methylation in the wild type. Here we have in the farm, in the early domesticated, and this is the X of the early domesticated. So the change, the lower values are already established very early. And this has differences, has consequences in the level of methylation. So hypermethylated in the wild, hypomethylated in the farm animals, and then you see changes in gene expression, concurrent changes. And this, the same thing for other genes. So what we conclude here is that the first steps of domestication involve mutations that are established in genes related to development processes and are expressed in the embryonic germ layers, including the neural crest. But then, when we don't look only at the very beginning, at the early domesticated, one of the generation, but we look at more than 20 years of domestication, 25 years of domestication, so several generations, what we found is that in some genes, for example, the cadherin family number 9, we see that in the wild type, there is this CG that is not methylated. In the early domesticated, this is methylated. And then when we get and we compare it with several generations of domestication, then this is not methylated because there is a transition between a C and a T, so a CT mutation. Okay, and this is not by chance. This is a permutation test show that this is not by chance. And what this shows, or what this tells us, is that if you change the environment very different from the environment that the animal is used to live, and you maintain for time enough this environment, then this eventually, this environmental change, the genetic change, can be canalized, associated with the genetic change. And this is the proof. So there was one person, probably you know, Etienne Benchen, who made this very nice paper talking about that epigenetically facilitated mutational assimilation. Epigenetics as a hack within inclusive evolutionary synthesis. So this is exactly what we found here, right? And so the environment induces epigenes or mutations, and then these epimutations eventually become assimilated as a genetic change. And furthermore, what we found is that places where this has happened coincide with genes under positive selection. We look at 7 different vertebrates, including the fox, the cat, the wolf, the duck, the horse, and so on. No? So everything seems to match. No? That what we found is like we induce epigenetic change. This epigenetic change becomes associated or assimilated as a genetic change, and then these changes coincide with regions or genes under positive selection in many domesticated. Okay, so. And this not only happens in animals, but also happens in us, in humans. So, but this is a process in which we talk about how this epigenetic change can contribute to the speciation in individuals. No? So here we have different populations here, and this is the speciation continuum. So this is the ancestral population. This is the new species. This is work by Niska from Cambridge University. There are different environments, and then what he calls epimutation spaces. The animals that are here eventually become, and this is represented by the darker ones, become assimilated as genetic changes. So first you have the epigenetic change, and then eventually canalize into the genetic change. And I was saying that this also applies to humans because nowadays, it's possible to infer even the epigenome of very old remains like the Neanderthals. That's why Svante Pavo won the Nobel Prize in biomedicine in 2022 for the discovery, for making possible the sequencing of very old genomes. So, and he published a paper in which they were comparing the epigenome of Neanderthals versus anatomically modern humans. And when we compare the changes that we see in the CBAS between the farm civars early domesticated with the anatomically modern humans, the changes from here to here, we see that there are some overlap. That this overlap again is not by chance, it's just significant. So it means that the early stages of domestication, they have a parallel not only between fish and different animals like the horse, the cow, or whatever, but even with humans. This is in context with nowadays in evolutionary biology called the self-domestication hypothesis in humans. And then there are some human phenotypes that are considered hyper-domesticated. One of them is Williams syndrome. Williams syndrome kids are kids that are associated with hypersociability, reduced aggression, like the wolves, like the foxes, like low aggression, a smaller teeth, smaller faces here, again, everything related, depigmentation and early sexual maturity. And when we compare this with the early domesticated civars with the Williams syndrome, but not here, like here, 1 or 2 generations, but more than generations, we see again a parallelism. So it seems that there is 2 lines running in parallel, in which the more domesticated you are, the more changes in parallel that way. And last thing that I wanted to say is that although epigenetics may look like Lamarckism, is not Lamarckism. And I have to say that because there is a lot of people, especially people that are even editors of journals and magazines and stuff like that, that they talk about this, and I don't like that. So I'll try to explain why I think, not just me, as many people, who epigenetics is not Lamarckism, although it looks like. So as you know, Lamarckism propose, or the typical example, that the inheritance of acquired characters, changes are acquired during the lifetime. And the typical example is that the giraffes, nowadays, by the way, we know that has nothing to do with the food, is about sexual competition, but it's a paper that recently came in Science. But he proposed that because they need to reach the higher leaves and so on, so this makes to have like longer necks. No? And Darwin said, no. With the natural selection, said changes are already present in the parents, and then natural selection selects what is the most adapted phenotype. So the differences between Lamarckism and epigenetics is that the environmentally induced changes, according to Lamarckism, is what he was saying, a positive reaction. And most of the changes, he always was talking about adapted changes. And what we know is that more often than anything else, that the genetic changes are maladaptive, okay? And then the mechanism, you of course could not know about the mechanisms. And the mechanism is like, okay, a vital impulse, but what is vital impulse? Now we know that this is due to a epigenetic mechanism, mutations. We can measure them, we can localize them in the genome, we know exactly everything about them. And then what is being inherited is the phenotype. And now what we know is that what is being inherited is a specific pattern of genetic changes. So to put it into context, if we have here environmental change, how we go from here to inherited trait, so let's call evolution. So Lamarck was saying that changed needs and behavior, not only the giraffes, he was making an example of the arms of the Smith and so forth. So the use of structures leads to this evolution, this change. No? Then came Darwin, and Darwin said, no, there is one existing genetic variation that through natural selection selects the most appropriate phenotypes. And then came Waddington, who's the father of the epigenetics, and mentioned that environmental change gives rise to epigenetic response, as we have seen in this talk. And then eventually, and this is the key point that needs to be much more studied. We cannot say that there is conclusive evidence, but it seems like that. Everything points to this way, that this genetic assimilation then through natural selection. So it's not incompatible with natural selection. Okay? And the 2 last slides it's to, as a recap of like some reflection or some thoughts, no, on how to study epigenetics, no. So the study of epigenetic modifications, the what, you can ask in different manners, no. How we do these studies? And we can do these studies, what I call the inquiry axis, no? How we study this. And we can do a single gene or canonical approach if we know exactly the phenotype that is associated with that gene, or we can go if we don't have a priori conceptions with the omic studies. And from this, we can use DNA methylation or histone modification or or whatever. And then another question is like the who. No? So what we study one single species, or we compare these epigenetic modifications in different species. So this is another axis of inquiry. Then we can think about the where, you know, in what conditions we study this. Where do we study? Under this environment, under this other environment. We study these conditions under standard conditions, or we push the animal to out of the zone of comfort or very extreme conditions. Okay, so it's another thing that we have to think about that. And another even axis is like when, no? So early development or late development. This is very important because, as I said, there is windows of sensitivity which give rise to very different outcomes. But as you can see here, I always like to frame everything in 6 questions. It's missing the most important one, no? Which is the last one. Why? Why we study epigenetics? And epigenetics is now an attempt to integrate complex evolutionary phenomena into what we know in Darwinism. So in Darwinism, this mind is modified by novel. This is Darwinism is characterized by variation, Malthusian competition, as we know, and natural selection. Then came the modern synthesis with the new concepts, mutation, population genetics, micro-macroevolution, and then inheritance and speciation. Nowadays, there are new aspects, multivariate selection, in the extended evolutionary synthesis that several proponents are trying to push forward, like Massimo Pilucci and other people. And epigenetic inheritance is one of them, no? Evo theory, plasticity accommodation, niche construction. So epigenetics is something that has its own right to be part of this extended evolutionary synthesis. But there are many open questions. The interactions among mechanisms, something that is still difficult to understand. Nowadays, we have techniques like MOFA to better help us to understand these mechanisms, how gene expression and definitely the phenotypes is regulated, the phylogenetic differences, things that apply to one taxon does not apply to the other, aspects of epigenetic inheritance, the environmental response and phenotypic plasticity, developing biomarkers. I talked yesterday about the biomarkers, today I didn't mention that, but there is a lot of interest nowadays in developing epigenetic biomarkers, for example, the ones to predict or to see the outcome of certain types of cancer, colon cancer, lung cancer, and myometrial cancer nowadays are basically used or based on epigenetic biomarkers, not genetic ones. And then, of course, the contribution to adaptive evolution. So with that, the take-home message. So epigenetic mechanisms can act as drivers to generate diversity. So we saw that these epigenetic mechanisms act together in complex and still not well understood ways. We see sources of epigenetic variation that includes genetics and intrinsic and extrinsic environmental cues and stochastic events mentioned before. Then the maladaptive environmentally induced mutations, epigenetic traps, how this can be a danger in nature. Remember the sex control, the environmentally induced and transmitted susceptibility to masculinization, or how we exploit this in farming of the animals, for example. And then the inheritance, legal reprogramming of DNA methylation in aquatic animals. This theoretically suggests a higher chance of genetic inheritance. But this inheritance comes into two flavors, no? The context dependent and germline dependent, depending on whether the germline is involved or not. And then we need more proof of the environmentally induced epigenetics changes transmitted to the offspring. And also that domestication is a very, very good place to where to investigate epigenetics. No? Epigenetic regulatory mechanisms, they play, we think, a central role in the process of domestication across vertebrates and seems to involve cells of the neural crest. No? But domestication is a very, very good place to where to study epigenetics. And finally, la raison d'être, no? The epigenetics should be ecology and evolution. I think that nowadays cannot and should not ignore the many contributions that epigenetics can bring about, enhancing the debate on the contribution to the extent of evolutionary synthesis. So in a group of in a place where the evolutionary biology studies, I think that epigenetics nowadays is something that that should be part of the program of research. And for my part, nothing else except to acknowledge the group, the members of my group, the work that I presented today is the work of my PhD students, Nuria, one of them here, and then collaborators and grants, and you for your attention. Thank you very much.
Thank you very much. We are already quite ahead of time. So maybe if there is one question maximum, or otherwise you can, of course, continue discussing after the seminary.
Yeah, one question. Thank you very much, Francis. It was very, very interesting. We want to talk about how one stress source that is applied one time can be inherited and maintained in population. Do you have an idea or data about repeated stress? I mean, how if the same stress that is not usual for the species is repeated years after years after years?
No, but we have now about to investigate the F2. So we have working with fish, and especially with the sea bass that takes so long to raise. It is a problem because it takes us 10 years to do an experiment, no? But using other systems like zebrafish or model species should be easier to track this question. This is something that should be done in many different generations and see what happens, and to have a clear. So I would do is to push the animals in a way that you modify them in such a way. I don't know, can be by temperature, can be by pollutants or whatever, and then try to see how many generations this lasts, because there is a lot of controversy. For example, I was mentioning yesterday that when they use C. elegans, the worm, they go up to 80 generations. But this is false because, and this is said not by me, by the people who does the experiments, because to do that, they use animals that are modified, genetically modified, transgenics, to then modify the temper with epigenetic mechanisms in such a way that then they can go for many generations. But when you use wild type C. elegans, then the effects go for 8 to 10 generations. And remember that C. elegans has a life cycle of 3 days. So how this would happen in a vertebrate or in invertebrate with longer life cycles, it's difficult. This is something that it should be another student of mine, Daphne Nastasiadi, has a review paper in which has a table or diagram showing like different generations, but always is a few generations. So I don't know what would be the effect of repeated exposures for many.
Thank you very much. Thank you very much all for being here and people online, and see you next week for the next seminary.
Thank you.
Thank you.